Hello, and welcome to today's webinar, Achieving High-Quality Spirometry Data in Hybrid and Decentralized Trials. Before we begin, I'd like to review some key audience tips to give you the best webcast experience. Today's webcast is being streamed through your device, so there is no dial-in number today. Please check your speaker volume to ensure it is turned on. And also, for the best webcast experience, please use Google Chrome. Be sure to enable Adobe Flash, the media driver, and we recommend closing all windows except for this browser. To change the size of the video feed and the presentation on your screen, drag the slide bar at the bottom of your screen to the left or right to adjust your screen size. Your control panel is on the bottom sidebar on your screen. The bottom red Leave button is how you will disconnect at the end of the webinar. If you have any technical difficulties, please use the chat box. We have relevant resources available for you today. You can access and download these resources within the chat box. Please provide your feedback about today's webinar via the survey immediately following the broadcast. Most important is the Q&A icon, where you can submit questions to the presenters at any time during the webinar. Please use this window for presenters and the moderator chat for technical difficulties. If we are unable to answer your question during the webinar, we will follow up with you separately, so please don't hesitate to ask questions. If you would like a copy of today's presentation, you can send a request through the moderator chat or later to marketing@ert.com. And finally, an on-demand version of today's webcast will be sent to you shortly after the webinar. So let's get started. I'd like to welcome Clario's guest speaker, Dr. Michael Beeh. He is the founder and medical director of INSEP Respiratory Research Institute, Wiesbaden, Germany. Joining Dr. Beeh are Kevin McCarthy, Director of Respiratory Solutions, and Dr. Phil Lake, Respiratory Solution Architect, both from Clario, and I'm Dawn Patterson, also Director of Respiratory Solution at Clario. So it's been a roller coaster ride these last few years for clinical trials. The pandemic has made everyone jump through hoops to make sure the clinical trial continues without any hiccups. Some of the questions are, how do we continue to collect good quality data during the pandemic? What are we going to do if the patients aren't able to come to the site? If they can't come to the site, well, then I guess we have to go to them. Are there any other ways to collect good quality data without going to the patient's home? Well, in today's webinar, we are going to discuss how to collect good quality data at home without increasing site and patient burden. Now, I'm not saying there aren't going to be challenges, because with change, there's always challenges. However, you will see and hear ways to make the transition from clinic to home a lot less cumbersome. There are several scenarios. We have the clinic data, hybrid collection, clinic at home, and with home collection, will you use healthcare providers, have self-coached or virtually coached, or both? Although collecting full spirometry, the FEV1 and FVC, is new at home, Clario has been training sites in coaching their patients to collect FEV1 and PEF for over 20 years. So we do have an understanding on what it takes to collect good quality data from home. So Phil, with all the data we've collected, what have we learned from collecting all of this data?
Yeah, thanks, Dawn. If we can move on to the next slide. I wanted to show you a little bit of data. So this comes from a study where we were collecting both home spirometry and clinic-based spirometry. And what you can see here on this chart really is really the correlation between handheld data at home and spirometry data in the clinic. Now, if you were to see a kind of a perfect correlation, you'd see this dotted red line in the middle. So all of the data points would fall along that line. Now, we don't see that, so that the best fit line for the data is actually the black dotted line that you can see here, which is actually shunted a little bit further to the right. Now, what this tells us is that the lung function generated at home is slightly lower on average than the clinic when you look at a population kind of level. And so there's a few kind of drivers for this. I mean, it's not totally unexpected, because if a patient's actually generating data at home with their own device, effectively they're just going to be following core instructions and actually taking a nice deep inhalation, blasting it out, and the device will feed back to them as to whether they've actually generated a good effort or not. Now, when they come into clinic, what you've got then is a fully trained healthcare professional whose job it is to make sure that the patient is taking optimal inhalation, so all the way up as far as they can, and then blasting the air out as hard and as fast as they can until they reach a plateau. So you can imagine, doing this yourself, you get a little bit of motivation, but if you've got your own personal trainer saying, go on, breathe in harder, blow out harder, you're always going to perform better. So it's the same with anything in life. So again, it's not unsurprising that we see better data in clinic than we do at home. Now, you will also notice on this chart is that there are a few outliers here. So some patients do significantly better in the clinic or significantly better in the home. And so this is kind of down in a little bit just the variability and technique. So as good as humans are, we're always going to have a little bit of variability for the individual technician coaching the patient. So we may see differences between the individual coaches at clinic. They may change over time. Or again, we do get variability in terms of diurnal variability with lung function and also with the patient's disease state. So it's not surprising that we see a little bit of variability there. But the core large levels of variability is often related to shortcomings in the guidance, such as ATS 2005. So the biggest driver of variability is suboptimal inhalation. So with ATS 2005, all of the focus was on how hard and fast you blasted that air out. And Kevin's going to explain a little bit about this in a few slides. But again, if you don't take that full breath in, effectively, you kind of really minimize the kind of value. And over time, if you then start to improve that through a kind of a core learning effect, you will alter the data. And this is one of the key problems that we face in clinical research. It's not just a case of taking a device and saying, well, this measures accurately. It's effort dependent, and it's all about controlling that variability will determine the kind of the approach you take as you go forwards within clinical trials.
Great, so, thanks. Yep. So what have you seen, Dr. Beeh, in your lab with patients using the AM3 to collect FEV1 and PEP?
Sorry. Well, thank you, Dawn, for bringing this up. This is, in fact, some very important issues we're discussing today. And I'm happy that Phil actually showed these data because it exactly mirrors my experience that I have from a situation in clinical trials, both doing on-site measurements, but also having some experience with data collection at home. It's not that patients aren't motivated. I mean, they are motivated, and you can see that there is this nice correlation between home collection and office collection. But of course, they lack this kind of supervision. And for them, it's just, well, if you do a good blow, well, that's a good blow. It's a maximum blow, and that's it. And they do not understand the implications of variability of data. And it's very important that they really try to get out the maximum of the maneuver. And for that purpose, it is, of course, as Phil already pointed out, always helpful to have a trainer there who understands the physiology of the maneuver, maybe also the physiology of the disease. And he can actually advise subjects, say, yes, you've done a good job, but maybe you can do better. And that's very important because that minimizes variability. So my impression is very much, as is shown in this slide, on average, home collected measurements tend to be slightly lower than in the clinic, because in my clinic, nobody leaves the office with a submaximal effort. I can guarantee for that.
That's great. That's awesome. Thank you. Thank you both. So as Phil and Dr. Beeh stated, the data collected at the site shows much better results than if the patient collected data at home. And it's clear that variability has a lot to do with the technique. Kevin, can you briefly discuss one of the main drivers of poor quality data?
Sure. Thank you, Dawn. If we could move to the next slide. What I want to show you here is these are three measurements done on, they're clinic-based measurements done on the same patient. And they're showing highly variable results. That is, they don't sort of track each other too much. And that can happen certainly with medication or with an exacerbation. You can see the numbers go up and down, particularly in asthma. But what's different here, this is an example of when there's a technique issue at the clinic-based measurement. The red arrows directing your attention to V3 there, the red arrow is labeled optimal. And what we're talking about there is what we've got highlighted between the two horizontal black bars there is the deep inspiration that's meant to precede the forced exhalation. And this is something that really wasn't addressed in the 2005 standards. There's really no guidance there. All the spirometry standards have always said the forced exhalation will start from full inflation, but it doesn't give you any guidance as to how do you get there. And if you look at the other two measurements here, you can see that the amount that they're taking in above a tidal inspiration is really small in V1, and it's non-existent in V2. In V2, the forced exhalation starts from a tidal inspiration. So really, it's not a correctly done maneuver. And this is kind of what we're now seeing with the 2019 standards. None of these efforts show something that was introduced in the 2019 standards. But before I show you that, let me just draw your attention to where these forced exhalations ended. That is, there's usually a sharp drop when they start the forced exhalation, and then it sort of flattens out to what we call a plateau. And notice how the plateaus in each one of these measurements really kind of overlap. This was a somewhat surprising finding when we started doing studies with the 2019 guidelines, is to find out that, even when it's done incorrectly, when it didn't start from full inflation, the measurement can demonstrate repeatability amongst the efforts. Mm-hmm. So if we could move to the next slide here, let me show you what the safeguard now is. This is a representation of what the spirometry maneuver should look like on a volume time tracing using the 2019 standards. So we see tidal breathing, just relaxed, normal breathing, a deep inspiration, and then we see it rising to a peak. Now that peak is something that the standards actually do tell you now. You should be looking at the patient and the volume time tracing, kind of like the same way you look out your front windshield and then look over at your side view mirror with just a quick glance of your eyes. And when the command is given to start the forced exhalation, it shouldn't be given until you see that the patient is obviously making an inspiratory effort, yet we see no further volume change. Then the forced exhalation starts, continues until you see a plateau, and that's the end of the forced exhalation, but it's not the end of the test. The new standards specify breathing in as rapidly as possible, back up to full inflation again. And that volume that they inspire is called the forced inspiratory vital capacity, the FIVC, which you can compare to the FVC they just exhaled. And if they match, you're pretty darn certain that forced exhalation really did start from full inflation. If the FIVC is larger, though, it tells you the opposite. It tells you that, wow, that didn't start from full inflation. So that's what we're seeing now in the 2019 studies.
Wonderful. Thanks, Kevin. So training a technician to coach that patient to full inflation will definitely decrease variability in the data. That's awesome. That's great. Now, we've collected data, the 2019 data. We have a few studies that are running right now, and we're collecting all this data. Can you give us a brief summary of our findings to date between the 2005 and the 2019?
Yeah, I'm happy to. So this slide summarizes what we took a look at a study utilizing the 2019 standards. It was looking at patients with asthma. And so we kind of took a look at what was good quality. We noticed that, it was very possible to do what we just saw, acceptable and repeatable efforts. They really didn't start from full inflation. So we looked for that. And what we found was that roughly 13% of all the measurements out of 700 in an ongoing study were acceptable and demonstrated repeatability, but failed the full inflation test. And this wasn't just a busy site where there was one or two unskilled operators. Far from it. What this is people who have developed a technique because these measurements came from about 50% of the operators from about 50% of the sites. And on average, they were starting forced exhalation from around a little under 300 ml below full inflation, but up to 722 ml, 3/4 of a liter. So clearly the operator isn't looking at the patient. They're not looking for that sweet spot as to when's the appropriate time. Rather, they're delivering the instructions in a cadence that yields repeatable results. So they're starting these on average from around 9% below what the actual FVC would be. And if we could take a look at the next slide, I have just a little bit and one other point that I wanted to make. We compared that study that I just told you about, that asthma study, with a completed asthma study that utilized the 2005 guidelines. And both studies had at baseline, and this is a good practice, 2 baseline measurements 30 minutes apart. And the effect of using the 2019 guidelines, and we believe that full inflation standard, caused about a 50% reduction in the variability in the FEV1 in those 2 measurements that were establishing a baseline. So really a much tighter baseline. And then if you looked at the coefficient of variation of the FEV1, which is really a measure of how variable is that from one measurement to the next, there is nearly a 50% reduction in that as well. So the 2019 guidelines seem to be having the desired effect of keeping data that really doesn't belong in the clinical trial because it didn't start from full inflation. That's the #1 instruction.
That's great. That's great. Thanks, Kevin. Thanks for that information.
Certainly.
So I mentioned at the beginning that there are several scenarios in collecting data. You have the clinic, the healthcare provider going to the patient's home, self and remote coaching. Let's discuss collecting home data with the healthcare provider, self and remote coaching. So can you discuss some of the learning effects?
Yeah, I think so. What Kevin is highlighted to us is, I think, is really critical information. So, and kind of sort of to summarize, I mean, when we're looking at ATS 2005 guidance, the site may well think that they've met all of the criteria for good, reliable data. But we've seen here in a percentage of cases, the inhalation is suboptimal. And the problem we have with some of that is that you often get a little bit of a learning effect as you run through the trial. So patients, don't like undertaking spirometry. And when they go through repeated spirometries, they gradually get better at generating this. So, and that kind of learning effect will actually confound your drug response. So if the patient's gradually generating higher and higher FEV1 with every visit, if they're on placebo, they then return into a responder patient. So, and that's going to undermine your ability. It's going to add to the overall variability of data, and it's going to undermine your ability to show the true treatment effect on study drug. Now, there's a couple of other things that you need to understand about this as well. So when we go into a kind of a remote setting, and so if we want to undertake spirometry outside of the clinic, in the clinic with ATS 2019, and with the site being fully aware of suboptimal inhalation, or if you happen to go to Kai's clinic and they're spot on already, you'll get optimal inhalation and a reliable baseline, and you can move forwards. As we go into this kind of digital trial setting, this whole area of variability is really in itself variable depending on the nature of the data collection you take. So if you're just going to go for patient-generated data, it's very difficult for patients to fully understand whether their inhalation is optimal or whether their data is optimal. You can have algorithms that pick up some of the ATS standards and feed it back to the patient, but the algorithms don't cover everything. I mean, they don't cover the posture, they don't cover the seal around the mouth. So there's still quite a lot of variability there, which is outside of any kind of standard feedback. And if you're feeding back to an untrained individual, they won't necessarily pick up exactly what they need to do to correct that and to get optimal data. So there's a few things you've got to understand then. So if you're going to go for a remote patient-generated data where the patient is coaching themselves, ideally you need to change the design of the study where you have a longer run-in, so some of those learning effects are picked up before you actually introduce your drug. Because you can't see a drug effect accurately if you've got a confounding learning effect going on at the same time. And we've seen that in some of the published data looking at digital handheld data, where at the beginning of the study, the ATS compliance is way worse than it is at the end of the study. And again, those studies have been published with ATS 2005, and we've already told you that ATS 2005 isn't actually picking up all the variability anyway. So even using ATS 2005 compliance as a rule of determining whether your home-based data is good or bad is fundamentally flawed. You need to look at the variability of the data and how that changes over time to really understand that. So that's the first point I wanted to make. Sorry, it's a bit of a long answer. The second point is that you then, if you're not going to go for the patient-generated data and you're going to try and minimize the variability that could generate, you've only got two options. So one is a visiting healthcare professional, and that can be expensive. So, and some of the data we've looked at with that, the best healthcare professionals in the visiting mode are just as good as the clinic-based professionals. The trouble you often have is that if you have a very broad range of studies, it's quite difficult to get really highly trained respiratory technicians to be that visiting healthcare professional. So as you become less experienced, you get more variability and you can introduce problems. The next way of doing it really is to have a remote visit from the site. Now, this really is got to be considered the gold standard, because you have highly trained individual at site with all of that kind of interaction with the patient, and they then can actually gauge all of those little nuances about variability and really drive better data quality. So hopefully that's broadly answered your answer. I'm sorry, your question. I'm sorry it was so long.
That's okay, Phil. Great job. Thanks for the information. Kai, what other considerations are needed when measurements are collected at home?
Well, I think basically the main principles should be exactly the same for this remote measurement at home like it is in the clinic. Because I think the whole charm of the idea is if we are able to transport the level of quality that we achieve in a clinic or a study site, we can transport this to the home environment and to the quality of remotely collected data, then we have a large opportunity for making lives easier, not only patients' lives, but also operators' lives. And I give you two everyday examples from my life as a clinical researcher. Now, we're doing a lot of respiratory studies, and you all know that there are date constraints, for example, in visits, but there are also time constraints, because we know that, for example, asthma has got a lot of circadian variation in lung function. And that quite often brings along a lot of problems, because I tell a subject, well, Mr. X or Mrs. Y, you not only have to come exactly on day 7, it cannot be day 6, it cannot be day 8, you have to be here on day 7. And to make it even worse, you have to be here just exactly between 7:30 in the morning and 8:30 in the morning. That, even aside from COVID, already is a very difficult situation. And a lot of recruitment is blocked by the difficulties of finding these appointments or matching the protocol. So that, of course, would be a very, very nice situation where you could say, okay, there is not necessarily the requirement to come up to the site. We could do this at home. And then we are much more flexible because you don't have to travel. You may even look for your children if they're at home and you cannot leave home and stuff like that. So even if you're afraid of COVID, you can stay in the house. So that's option number one, or aspect number one. The second aspect, something which is also quite often overlooked and forgotten, is that we usually measure lung function after washout of maintenance therapy or rescue medication. And on paper, that sounds easy, but for many patients, that's quite difficult. You get up in the morning, you're not allowed to take your background medication, and then you have to travel to a site. This can be 1 hour, can be 2 hours, and then finally there you get your lung function, and then after the lung function, you're allowed to take your maintenance therapy. Now, I can tell you that many patients quite often secretly take rescue medication. And you know that this adds not only variability, but also, I mean, this jeopardizes the credibility of your data. If you, for like a patient like that, if you're able to say, well, we can do it more or less directly at home, you get up in your home environment, we do the remote supervised measurement, and then you take your rescue medication, that's far easier for the patient. So these are two typical aspects of clinical trial where I think it would be very helpful to have something like a high quality remote collection of home spirometry data.
Dr. Beeh, that sounds great. It really does. Thank you very much. Thank you, Phil. Collecting data, whether it is in the clinic or at home, of course, the main goal is to make sure that it not only meets the ATS-ERS criteria, but that data variability is decreased. Clario has put together a solution to collect good quality data while the technician is at the site and the patient is home. The patient doesn't need a computer just the sensor and Clario's iSpiro app. So I'm going to show you a demo of a remote video call where it will display real-time spirometry collection that you can actively manage. You're going to be able to see both screens. You're able to see the subject's effort as well as the patients themselves. Looking at their posture, making sure they have a good seal around their mouth, around the mouthpiece. Now, the first 30 seconds, you will see our portal, and this is where the technician will start the session by, and then following by a screen showing both the technician and the patient. So, the demo.
So I go to the study portal and I see I have a study open. My site has several different patients, and here I have my scheduled events, and I see there's an event upcoming. So I click here on start to get in contact with my patient. It's loading, takes a moment. Now the camera shows me sitting here in my office. And here is my patient, Jan. Hello, how are you?
I'm good. How are you?
All right, very good. So, we have a spirometry scheduled today. You are well instructed about the maneuver, so we don't cover the whole measurement phases. So let me start the event. You have the sensor with you?
I have.
Okay, very good.
Nose clips.
And nose clips. Okay, I start the event, put the nose clips on. And now, as we discussed, put the sensor in your mouth and do a few normal breaths. And then when I give you the command, after 3 or 4 normal breathings, one more normal breath. Okay, now big breath in all the way up, 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 biggest breath, and blah! Push, 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 push. Keep going, keep going, keep going, keep going until you're empty. A little more, a little more. You can do it, you can do it. All right. Then at the end, very good. All right. Okay. So here I see the live data that Jan has recorded at his home. I see there was an error, a little bit of a problem during the start of the test, and the end of the test, the plateau was not completely reached. So Jan, you need to do a little better job this time. Don't wait too long. When you're completely full and you cannot inhale anymore, blast it out as soon as you can. Okay? And then keep going until you're really empty and until I tell you to take another big breath in. All right?
So as the demo showed, collecting the data in real time while the patient is at home can produce good quality results. Clearly, you can see the patient, their posture, make sure they have a nice good seal. So that's really important to make sure that the data quality it doesn't have a lot of variability. However, there are certain things that should be considered. So Kai, what should the site and patient consider if they're collecting spirometry at home?
Well, Dawn, first of all, I have to say it looks awesome. I'm really thrilled by this demonstration because there are different aspects to it. First of all, it looks exactly as the spirometry in my office. So I really feel at home when I see this type of screen. I can see the flow volume tracing. I can see the time volume tracing. So everything, I'm in spirometry heaven, if you like. So I can see everything that I need, and I can also see the patient. And the latency is just great. The latency between the patient breathing and what is on my screen. Second, that's an important point. The latency is great. And third, I can have a look at the patient and at the graphs at the same time. That's even better than it is in my office. So I have got real-time data about performance of spirometry. And as you've seen in this wonderful example, I was just about to jump in because when he started with the inhalation, I was to say, hey, I saw a little bit of back extrapolation there. And then finally I thought, oh, whoa, that's not a fine expiratory plateau. And that's exactly the point, because no way would you have picked up these two errors, which you have beautifully picked up with your software device, but also with your technician, But no, there's no way that you would have picked up these two critical errors in a just self-collected environment with the patient self-trained. So really, for me, this is a very, very nice solution, and I'd love to work with it. But of course, I mean, like with every remote or technical solution, I think it is very important. You have to keep the patient in the comfort zone. That is absolutely critical, which means connectivity is absolutely key. So the technical aspects should be easy. Connectivity should be easy because that keeps patients at ease. And of course, ideally, he would be trained by an operator that he knows, maybe a guy from the site or a lady from the site. So he is really in his comfort zone. He's not having this stress of, am I doing anything wrong? Am I wasting my operator's time? Something like that. That's very critical. If you guarantee like that, connectivity, well-trained personnel, maybe the personal touch with trainer and subject, and together in a technical environment like this, I think you've done really, or you've created a really near perfect space for collecting supervised remote data for spirometry.
Yeah, I mean, there's one point I wanted to add to that, Dawn. I mean, for me, it's almost like a head-up display in a car. You know, you can actually look through the windscreen and see the dials at the same time. But so I think that the critical thing for this, and the main problem we have with this suboptimal inhalation, is it is actually quite difficult to look at the patient and look at the flows, the screen at the same time, to work out whether you're at the top of peak or not. And I think what we've often seen, technicians do to get around that is they go through the process. So they go deep breath in and then blast out, and they do it at the same time with the same instruction consistently. And it's not necessarily at full inhalation. And this is one of the reasons why I think with ATS 2005 is that you had highly reproducible data that was not necessarily optimal. And so I think this kind of head-up display approach is really going to help you, address that. So it's very exciting.
Yeah, it is. And if the protocol calls for 2019 guidelines and home collection, I guess we can all agree that the remote coaching would be the best option for that protocol, right? All right. So decentralized clinical trials, they are the future. They're not going anywhere. It's convenient for those patients that live hours away. It's also critical for patient enrollment and retention, especially those with rare diseases. Having the right tools will optimize the site and patient's experience. So with Clario's remote coaching, the site can coach and ensure the patient is performing the spirometry measurements correctly and receives advice on how to improve on the next effort right in the comfort of their own home, which is wonderful. We've had quite a few questions come in so far. As a reminder, you can submit your questions using the Q&A icon at any time. So the first question, patient burden is important. How do you ensure the patient is not overwhelmed when collecting the data?
That's a good question. Yeah, that's a very good question. So I think this is one of the critical things that we see. I mean, it's it's a highly effort dependent approach, and you're expecting an awful lot from patients. So I have seen sponsors say to me before, well, does it really matter? I mean, we've been doing this for years. We've been getting patients to collect this twice a day, morning and evening. Why don't we just do the same thing with these new devices? The key difference here, and the one thing I'd really love to stress to anyone thinking about this, is that historically, what the patients were doing was generally PEF. So short, sharp inhalation, quick blast out, and then we were getting PEF and FEV1 off that data. It wasn't ATS compliant full flow volume loop spirometry. So they weren't doing tidal breathing, a good strong inhalation right away to the top and blasting out. And we weren't expecting 3 technically acceptable and 2 reproducible efforts. And as you know, in clinic, we often see on average about 4 efforts per session to try and meet ATS criteria. So if you're thinking about the burden on the patient, I mean, I don't believe anyone has ever asked them to do full ATS compliant spirometry once a day, let alone twice a day. And so you've really got to consider how tiring this is and what kind of time commitment it actually takes up. So, so I think we need to really kind of think about the patient in terms of how we balance that. I don't know if Kai or Kevin, you want to comment.
Yeah, I would absolutely, I would agree with that. A patient burden is clearly a critical issue. And of course, I mean, you have to take into consideration what phase of clinical trial you're operating in. I think, of course, in phases 2 studies, for example, lower numbers of patients, then it is possible to select maybe the most compliant population, because you really want to reduce the noise to signal ratio to the maximum. These are all, as we know, phase 2 studies. They are all more or less controlled, tightly controlled experiments. And then, of course, once you move to phase 3 and then maybe phase 4, real life studies, then of course you have to always balance between patient burden and achieving the best possible data quality. And as you mentioned, of course, repeated attempts of full ATS compliance spirometries multiple times a day can, of course, overwhelm patients. So I think it is very important to always compromise on that. So what level of quality do I want to achieve on one hand, and how far can I go with that without risking to lose data because patients get frustrated and their compliance goes down? Because if you say, okay, you have to do this 5 times a day, ATS 8 efforts or something like that, you end up with, I don't know, whatever, 20% compliance in the end, which leaves you with 80% of missing data. And then again, you have a different problem in terms of analysis and statistics. So I think this way of balancing quality and practicalities is something which, of course, we need to learn and elaborate in the future. But that's one of the reasons why we're here and discussing these new techniques.
Exactly. Thank you. Here's another one. This is a good one. All right. Are there any regulatory considerations regarding remotely coached spirometry? How do they feel about data collected this way? Who wants to take a stab at that?
I'll take the first crack at it.
Thanks, Phil.
That's another very I don't know if it's the same person, but they're spot on the mark here. So yeah, so this is again a kind of highly complicated area. So the FDA have actually started kind of reaching out with a kind of an advisory document or a request for feedback on their advisory document. And the key thing here is around validation. So again, you can't just take a sensor and show that it's kind of accurate at recording lung function and then just assume that that's it, and that everything else is going to follow. What you need to do is actually show that by moving into this kind of digital space, that you're still able to pick up drug effect as you would be doing in clinic, because there's a lot of variables there that really control the nature and the variability of your data. And so I think what Clario are doing at the moment is embarking on a whole series of validation studies looking at the variability and quality of the data. So you can't just look at ATS compliance for the reasons I've mentioned before. So that's not going to prove very much at all. What you need to do is actually understand how each of the modalities affects the variability of data and your ability to show drug effect. The other thing you need to do as well as part of that validation is to understand that the approach is fit for purpose. So and again, this comes down to what the FDA are looking for. So, I think it would be completely inappropriate to expect a patient, as we've just mentioned, to do full ATS compliance spirometry once a day for a whole period. It's just going to drive variability, and you're going to get massive kind of withdrawal because patients just can't cope with it. So, what we really need to understand is that, what balance. Each of these different modalities drives different variability and will have a different ability to pick up treatment response. And you really need to tweak the design according to the phase of study that you're generating. And so, what I think part of the validation approach will allow us to understand is that, do you go for one measurement once a day, or do you go for ATS compliant measurements once a week, or do you have a combination or something in between? And I think we're undertaking a whole series of studies at the moment, and I think at the end of that, we'll be able to understand and advise companies more authoritatively about what the best modality is to fit their requirements. But it's not just a case of getting a sensor, putting a Bluetooth onto a diary, and then recording it, once a day, twice a day, or whatever. So it's way more complicated than that.
That's exactly right. Any other thoughts on the FDA?
If I may add, and I think it's very important in this type of validation process to involve patients, because their feedback is very critical here. So what is the amount of burden they're willing to undertake in such a participation of a clinical trial? So I think that's a very, very critical thing here. Ask patients and ask them for their opinion and let them find out what would be their optimal balance between data quality and burden on the other hand.
Yeah. And the other thing I'd like to mention briefly is that you've got to be very careful when you do this. So, I mean, we could potentially design a study that would give you, perfect lung function data quality, excluding a whole range of patients who aren't very good at the technique. But again, the thing you've got to be very careful about is that, we've got an expression, you don't throw the baby out with the bathwater. So, I mean, what you don't want to do is exclude patients with more variable lung function, because they may actually be those that are most in need of the therapeutic intervention and may show the biggest therapeutic intervention. So it's not just about lung function. You've got quality of life and disease control and the rest of the core endpoints. So, you have to understand what happens when you actually modify the design to all those other endpoints. It's not just, FEV1 as a single endpoint. So you really do need to understand what you're doing and what impact you're having on the whole study holistically. So you need to know what you're doing. I think is the core message there.
Thanks, Bill. Thanks, Dr. Beeh. Kevin, what are your thoughts about collecting 2019 guidelines with the self-coached approach?
I think that's an ambitious reach. I think that we're not there yet. There are certain points during the forced vital capacity maneuver where there's the natural human tendency to avoid discomfort. Example, going up to full inflation. We've highlighted the importance of that and reducing variability, yet we're not telling you how it feels. And if our audience members, any of them have ever experienced spirometry and they've been coached to full inflation, they know that last 5% isn't really all that comfortable. And then that's followed immediately afterwards by that sharp, forceful exhalation, as hard as you can go. And that's maintained until you're as empty as you can be, even beyond where you feel like you're empty. And so those are the points during the maneuver, getting to full inflation, getting a really emphatic hard blast, and then emptying till you really are satisfying the end of the forced exhalation criteria. Those are points where the patient's really on their own. They're really not going to, it's predictable. They're not going to do as well.
Right. Thanks. Thank you. Okay. So Kevin, I think this one's going to be for you as well. From an overread perspective, if you apply the 2019 guidelines, of course, there's going to be a decrease in variability, but where is the change from the grading? At what point does it affect variability?
It really crosses all of the lines of the grades, the A through D and F grades that they cited in those standards. The data that I was showing you were A quality, A and B quality, that 13% of all of the measurements were A or B quality by the 2005 criteria, even though we weren't grading it by then, but it was acceptable start, acceptable end, and repeatable. And yet they didn't start from full inflation. And in the 2019 guidelines, that's no good. And it's, that was to make a point that, even what we thought was the best quality data can suffer from the submaximal inhalation error. And certainly the C, D, and F quality data is susceptible to that as well.
All right, great, thank you. Well, unfortunately, we do not have time for any more questions, but the team will follow up with those of you whose questions we're not able to answer today. Thanks to our presenters today for sharing your insights on today's topic, and thank you, our audience, for joining us. Just a reminder to please take a moment to complete our survey and share your feedback on our website. Thank you and enjoy the rest of your day.